Assalamualaikum warahmatullahi wabarakatuh and a very good afternoon to all of you. Welcome to INBIOSIS webinar lecture series for 2022. I hope the video that we played just now has provided you with some background of INBIOSIS. For those who are interested to collaborate with us, you are most welcome to do so. So we will also organize many workshops and activity for this year. Feel free to browse INBIOSIS website and social media for the latest updates and info. I feel so grateful today that all of us are granted good health and time to attend webinar series. So far, this is our fourth webinar for this year. There will be many more webinars to come, and I hope all of you will join us again in the future. For an introduction, INBIOSIS webinar series is established to provide knowledge and research experience sharing platform to ensure we are still connected with each other and stay motivated in our research despite this sudden pandemic COVID-19. So without further ado, I would like to introduce our honorable speaker for today, Associate Professor Dr. Syarul Nataqain Baharum from INBIOSIS. So allow me to share Dr. Syarul CV. Dr. Baharum graduated from University Putra Malaysia. After joining INBIOSIS in 2008, she developed and received her postdoctoral training in the field of metabolomics at the University of Sheffield, UK. Currently, Dr. Baharum research is focused on the new insight of analytical and biological perspective of the metabolomic in the field of system biology. It is particularly aimed at the understanding of secondary metabolites production in local herbs plant, especially on polygonominous, which is kesum, and Metragyna speciosa, which is ketum, as well as fluxomic study of Lactococcus lactis. She has expanded her research focus in disease study of grouper fish and also Alzheimer's disease in human. Currently, she has published more than 100 research articles, 4 books and 6 chapters in books, and more than 30 invitations as speaker in local and international conferences in metabolomic and functional metabolomic related field. She also received more than 4 million ringgit research grant in total from MOSTI, MOHE, and industries. Under her supervision, 9 PhD students and 18 master students have graduated. And currently, she is supervising a total number of 21 PhD students and 18 master students as main and co-supervisor. As principal investigator, she has filed 2 patents and granted 1 trademark and 1 trade secret. Her work has been awarded prestigious including Bio Innovation Award 2011, Selangor My Innovation Award 2014 and Innodex 2021. She also was awarded as visiting lecturer to Chulalongkorn University, Thailand 2017. She played a leading role as Malaysia representative in Asia Oceania Metabolomic Forum, AOMF. She also engaged in collaborative partner with industry such as FGV Sdn Bhd, AAR Sdn Bhd, Live at NES Malaysia Sdn Bhd, Narina Group and Medica Natura Sdn Bhd to take a further step to commercialization. Currently, Dr. Baharum is a principal investigator of Metabolomic Research Group and Deputy Director at Institute of System Biology, UKM. Okay, let us welcome Associate Professor Dr. Syarul Nataqain to share her research finding. The floor is yours, Doctor.
Okay, okay. Thank you, Dr. Muni, for the kind introduction. So, thank you for INBIOSIS and also the webinar team for inviting me to be a speaker for this session. So, I would like to share with all of you on my current research in metabolomics field. I hope that from the research that we have established can give some insight to what is the trend of metabolomics, especially in Malaysia. Okay, as a title of my presentation, an emerging technology for the sustainable agriculture. So in this talk, I will be focusing on 2 main of my research project. One is on the plant, another one is on the animal. Okay, before I start with that, so I would just like to share with you on the publication trends uh, since 2000 to 2018 because our book was published in 2018. So this is a trend on the metabolomics. When you use the keyword of metabolomics, okay, you can see the trend is increasing, okay, from 2000 up to 2018. I'm sure up to now there is emerging of metabolomics field and is increasing year by year. Okay, so as you can see here, the work has been published in very high impact journal. For example, the Frontier is Plant Science, which is the top 10% journals, and Bioanalysis, Anticancer, Acta, and so on. So this is something that we should look into and also take this opportunity to have more work on the metabolomics so we can publish in very good journals. Okay, so before I start with my research topic, so it's just like to maybe just to give a brief on the what is a metabolome is all about. Okay, metabolomics is the multi-parallel measurement of the level of large number of cellular metabolites, typical several hundreds or maybe thousands, and it's not many has been identified. What you can see is just a peaks in the profile, okay, and then you know nothing about that compound, okay. That's the challenge of metabolomics, okay. So you can see from here, from the center of dogma, you can see that from the genomics, proteomic, then we know that transcriptomics, and now is metabolomic is come in to complete the system biology of on the organism of study. Okay, so based on our previous recent publication, so would like to see how the metabolomic can be used as part of the omics multi-omics technology in the system biology research. Okay. So before I start, I would like to just highlight on the application of metabolomics approach in aquaculture. Okay, this is like a very useful tools because the metabolite respond in both natural and the stress condition in the environment. So it also to discover the biomarker profile of certain diseases that you have studied. Okay, more sensitive, more accurate and rapid technique. And it's benefit to our culture industry because we can assess the health status of the fish, the disease, and also the biological challenges. So this is one of the main project that I have in my lab. Okay, is on the metabolomic as a biomarker discovery in a fish disease study. So in this study, actually, we are focusing on the Fuscoglutatus, Ephenophilus fuscoglutatus, which is we call it grouper. Okay, why grouper is very important? Okay, as you can see here, it represents about 30% of the global marine fish production. And Malaysia is one as Asia main producer of grouper because this fish has been exported into Japan, Hong Kong, and Singapore, and the price will be very expensive especially during the Chinese New Year because of the demand of this fish. Okay, up to now, the price of the grouper is about 80 to 100 ringgit per kg in Malaysia, and it's much more expensive in Hong Kong and China. So if you are working with grouper, then you will become very rich. Okay, so fishery sector in Malaysia, okay, what is the production? Okay, it's about like 1.7 million tons in 2017, and it become increasing. Okay, with the total sale about 33,000 millions. Okay, the total sales of the production of the fisheries, especially in Malaysia. So as I mentioned before, grouper is contributing to 30% of the marine production. Okay, so one of the main famous grouper that been exported is Ephenophilus fuscoglutatus, which is the one that we study now, okay, and then Ephenophilus coides and then this another Ephenophilus, which is the giant grouper. Okay, so you can see that's why people are looking at the grouper because have very nice and nice taste with the very nice structure, the texture, I mean, okay? So you can have your grouper during your dinner today, maybe? Okay, so the sale of the, as I mentioned, the sale of the grouper is about 100 to 120 per kilogram. So it's become more the highest market value, okay, in the marine fish, especially in China and Hong Kong. So, but however, this production of grouper has been hindered by the disease problem. Okay, one of the main pathogen is bacteria. Okay, so it's contribute to the about 40, more than 50% of the disease problem. Okay, and then the mortality rate of the viruses, especially because viruses from is a bacteria. Okay, is up to 80%. Okay, affected the culture population. So this is something big loss to the grouper industry. Okay, so so one of the main problem is the fish disease and mortality. Okay, about 70% as compared to other problem. Okay, so that's why in our research group we have studied grouper for many years. Okay, one of the idea is try to see from the metabolomics perspective how the disease can be overcome. Okay, so from here we have using a different oil mix, but in this presentation I'll just share on the metabolomics. Okay, so we can see here from the fish disease. Okay, there's many way of the management of the disease. Okay, by using the vaccine, the antibiotic, and the pesticide, probiotic, and so on. And then as well on the feeding, which is the immunostimulants. Okay, but what is the what actually there's no concrete answer how actually to solve the problem, especially in Malaysia. So that's why our research group come in to be to try to solve the problem of the fish disease by using the metabolomics approach. Okay, so basically from our previous work. My PhD student Maya, she's working on this disease study of grouper. Okay, so based on her previous study, okay, this is the work has been published in as a review paper. Okay, so shows that from our research group, we are focusing on the Aphaniophilus proscocutatus. Okay, we have find out. Okay, the main finding of the research is the amino acids and also the fatty acids are contributing to the survival rate of the fish. Okay, so from here we have come up with the biomarker which is focusing on the fatty acid as well as the amino acid as part of our feeding formulation. Okay, to overcome the disease problem by feeding or immunostimulant the fish by using the feed formulation. Okay, based on this publication, this is like giving us a motivation how we can improve the study by come up with that solution on the production of the fish feed. Okay, by using the omega-9. Okay, part of that our formulation. Okay, because from our previous work, it shows that as compared to omega-3 and omega-6, it shows that omega-9 have give a prominent or remarkable result as compared to other omega. Our study is a bit different from the study has been published before because the other study comparing between the omega-3 and omega-6 only, but the study has shows that omega-9 is not giving any effect to the marine fish they are study. But in our case, omega-9 giving a very remarkable effect as compared to omega-3 and omega-9. That's why now in our study now we are using oleic acid as one of the formulation for the fish feeding. Okay, just to show to you, in a lipidomic study that we have come up with by using the lipidomic gateway, so you can see there is the pathway. Okay, we have mapping into the pathway. Okay, to see that how this fatty acid can be contributed to the survival rate of the fish. Okay, that because this is not only it's not only one single compound working, okay, but it's a synergistic effect that working together. But in our case, we have the omega-9, okay, that working very well, okay, to make that the fish that we study survive. Okay, so as I can show to you, this is the pathway of omega-9. Okay, all that the red color means that the high intensity of the metabolites, okay? So for example, the stearic acid, arachidic acid, and so on, that shows that uh, the fish has survived because of this increasing of these fatty acids in their body, okay? So based on that, we also study on the effect of amino acid and fatty acid. So we as what that we expected, amino acid and also contribute into the survival rate of that fish that we have studied. Okay, the one that we have infected with the vibrosis. Okay, so this is the pathway. If some of you attend my workshop yesterday, as I always said, that's the ultimate aim of metabolomics. Okay, is to have the biochemical pathway analysis for in order for you to answer the biological question and also to interpret your data, okay, your result. So this is the pathway that we come up with to show that how these metabolite are contribute to the survival rate of the fish. Okay, so I just like to demonstrate by using the LCMS stuff, we are comparing between the survivor and the infected fish. It shows that there is two different cluster, okay, between the control and also the survival fish. Okay, meaning that there is a different contribution of metabolites to the survivor and also to the infected fish. As you can see from the heat map here, okay, the metabolite that distribute in the grouper fish, okay. So for example, this is the metabolite for the control, okay, and then there's a metabolite for the one that has been infected with the disease, which is the survivor one. Okay, so you can see here most of the metabolite high intensity in the control group, okay, as compared to the infected and survived group. Okay, you can see that, meaning that most of these metabolites are not produced during the survival of this fish. Okay, because most of these metabolites are actually is used for the growth. Okay, for the growth, for the energy of the fish. Because what we did is we study the larvae. Okay, the larvae size, which is that the larvae size they still need a lot of amino acid, for example, for their survival, right? But after it's been infected, most of this amino acid is not is produced, but it may be in a very small amount, a minute amount as compared to the control one, the healthy fish one. Okay, but you can see here there is also some high intensity of metabolite also produced in the infected and survived group. Okay, for example, the some of the sugar group, yeah, some of the carbohydrate group, and so on. So meaning that they use this for the energy, for their metabolism, and so on. Okay. Okay, so just like to show you on the metabolite pathway analysis as compared to the in the survivor infected grouper. Okay, as the biosynthetic pathway of the immune response of the survivor grouper. Okay, you can see that's many of that amino acid and also are increasing. Okay, as like for example, like the aspartic acid, asparagine, okay, been increasing during that the fish is try to survive. Okay. Okay, so we can see here from this I'm not on the fish study, on the feeding study, we are still working on it. Okay, the work has been submitted to the journals. Okay, later once it's published, then I can share with you. Okay, but for now, what I would like to tell to all of the audience, okay, the aquaculture could feed the world and protect the planet if we get it right. Meaning that we need to manage how to manage the disease problem, okay, by using the different tools. In our case, we are using the metabolomics tools to understand what is the metabolite they are respond to the disease and how to overcome this, okay, by using the different techniques, okay. Okay, the second study is on the application of metabolomics in unraveling the potential of local herb. So maybe most of you know that INBIOSIS has been working with Kesum for many years. Okay, when I start my my work at INBIOSIS also, I'm working on Kesum. Okay, so we as one of the tasks for me that time is to do the metabolomics of Kesum. Okay, so okay, why we study Kesum? Okay, many of you may be saying that, okay, Kesum, so it's just a very cheap plant. You can get it everywhere from the market, and maybe you just get it from your backyard and so on. Okay. Why we study Kesum? Okay, as we know that Kesum have very pungent smell. Okay, they have very pungent smell. They have produce a lot of volatile compounds that make it interesting to study. Okay, so yeah, I know that you say, okay, you can get it in your laksa, on your asam pedas, and so on. Okay, that's why Kesum, because of this pungent smell and this pungent taste, make your laksa or asam pedas become more delicious, right? Without Kesum, if you make laksa or you make asam pedas without Kesum, you cannot make it, right? So you need that Kesum. So, okay, why we study Kesum? Because of this abundance of the oil glands, or we can say it's an abundance of the essential oil. Okay, Kesum produce a lot of essential oil. Okay, it's also natural source of aliphatic aldehyde. So it's been used for the food additive and also in the perfumery industry because they have very nice smell. Okay, and also nice taste. Okay. And also it's been used in postnatal tonic and treatment for the digestion. This is for the old folk people, okay, from the one that from kampong, from the village. During last time, they use this as a postnatal tonics, okay, and then for digestion. Okay, it also can have a digestive enhancing, anti-ulcer and anti-inflammatory, and also cognitive, okay. So people use it because of the smell and because of the pungent taste, okay, in their dishes, okay. So in the pipeline of the metabolomic study, okay, as I mentioned that there is a volatile compound, a lot of volatile compound in Kesum, right? But in our study, we don't want to leave the non-volatile one. We still want to keep everything. Okay, we want to profile everything, the information from Kesum, why Kesum why Kesum is so interesting to study, why they have produced a lot of this compound that have that benefiting human health. Okay, so in this study, we focusing on volatile and non-volatile by using the different approach of metabolomics tools. For example, GC-TOF, GC olfactory, GCMS, LCMS, HPLC, NMR. Then we come up with a powerful technique, powerful statistical analysis by using different multivariate analysis and visualization by using heat map and so on. So we want to have the comprehensive study of metabolite profiling of Kesum. Okay, so when we start the study, we realized that Kesum also can grow in the low temperature. Okay, it can grow in Cameron Highland, it can grow in Fraser Hill, and also it can grow in Genting Highland. Okay, there's a lot of Kesum there. If you go there, if you have time to go there, you can see in the area that the wet area, there's a lot of water there. You can see some Kesum grow there. Very nice, very big leaf and bigger leaf as compared to the one that we have here in this area. Okay, I think because they like very cool temperature, they like a very pleasant temperature there. Okay, so so once we find out that this Kesum can grow there as well, okay, so we want to study the population. Okay, what is it metabolite contribute to the differences of the population, or maybe because of the environmental factor and so on. Okay, so what we did, we grow the Kesum, okay, at the different temperature, baseline temperature, meaning that we put all different population of Kesum in one baseline, same temperature, everything same. And then we have the highland one. So, and then we also have the lowland one. Okay. So then we mimic the condition in the growth chamber. So because we cannot control, okay, as I said, we cannot control the environmental factor, because metabolomics, okay, the metabolite is very much influenced, okay, by the environment factor. Okay, so that's why we keep it in the growth chamber, then we control the temperature, the humidity, everything, so it will follow us. Okay, it not follow, then we can limit all the variable from the environmental factor. Okay, so from the study, we have managed to profile the flavonoid and also the volatile compound, okay, from the kesum, okay, by using our system in place at INBIOSIS. We have all the system, okay. So it shows that, okay, interestingly, it shows that all these plant, okay, are cluster, okay, cluster in the class to their population, okay. So we already anticipate this result, right? Okay, we know that, okay, like Lengging and Uluyang cluster together in the low population because they have a bit higher temperature, and Highland plant is clustered in the same group, Genting Highland, Fraser Hill, and Cameron Highland, because they have a low temperature. Okay. And then from the study in the growth chamber, okay, we find out that from different temperature study, baseline temperature, okay, remember the one that I put everything in the same baseline, okay, and then we have the higher temperature, and then we have the lower temperature. Okay, you can see that there is a biomarker that differentiate them, okay, based on their temperature, okay, the treatment given to them. Okay, for example, baseline temperature. Okay, this is the most the significant compound produced, dimethylcalcone. Okay, and also for the lower temperature, hydrocoumarin, and at the higher temperature, galactocatechin. Okay, so this is the compound, is a signature compound that produced at different temperature given. Okay. It's not moving. Wait, why it's not moving? Okay. My slide is not moving. Wait. Dr. Moni?
Yes, yes.
My slide is not moving, right? I cannot move the slide. I'm not sure why. Wait.
Can you start to move back forward?
Okay, I move upward and back. Cannot. Sorry for that.
Okay, maybe you what you can do, you can stop sharing and you can share back.
Okay. Sharing. Okay. Check the slide. Okay. Okay, now it's moving. Okay.
Okay.
Okay, thank you, Dr. Moni. Okay, so okay, and then for the volatile compound, okay, the one that I mentioned before is for the flavonoid compound, the non-volatile one. For this one is the volatile compound. Okay, based on the treatment given, you can see that the biomarker, okay, the signature compound from the baseline is the chirophyllene, okay, beta chirophyllene, and then we have higher temperature, which is we have the dodecanol, okay, the aldehyde. As I mentioned, decanol and dodecanol is the most abundant compound of aldehyde in kesum. Okay, and then we have the lower temperature. We have the signature compound, which is decanol and farnesene. Okay, so it's produced at different temperature given. Okay, so in meaning that, okay, as in order for us to manipulate, okay, to produce more of that kind of compound. For example, chirophyllene. Chirophyllene is very useful for the study for the insect repellent, for example, okay, for the some of the anti-cancer, okay, and then and so on. Okay, you can manipulate the production of this compound so you can produce more as you want, okay, by using the different temperature treatment. Okay. So, based on the different temperature study, okay, it shows that the cold temperature, the hot temperature, and the baseline temperature are clustered among their own group. Okay, so this is basically all the population has been treated according to their temperature. So all the population have been treated to the temperature, and then they follow the cluster based on their treated temperature. Okay, so okay. So, and then another study that we have come up with is like on the sensory, okay, evaluation of the flavor and also fragrance for the industry. As we know that Kesum have a lot of essential oil, okay, that can be used in the flavor and fragrance industry, okay. So that's why we come up with this sensory study, and then we have the system in our place, the GCMS with the INOS, okay, the INOS, okay. So we use this INOS to smell the compound. At the same time, we can see the profile of the compound in our GCMS system, okay. We can see the peak, at the same time, we can smell it, okay. So then you know, okay, this compound, like for carotene, this is the smell. Okay, and then for example, the dodecanol, this is the smell, okay. Okay, so because of this paper, one of the company, the one that produced the fragrance, okay, company contacting me to know more about the essential oil of Kesum in order for them to come out with the formulation. Okay. So okay, based from this study, actually we know that okay, the perfume when they make the perfume, okay, for the industry, the manufacturer normally will have 3 different notes, right? Okay, the first note will be the very nice very not nice or very strong smell, okay, the very strong smell, and but the strong smell is not last longer, okay. So that's what we have in Kesum. Eh, okay, that's what we have in Kesum. Okay, Kesum have the first note smell, which is the a bit because it have a pungent taste, pungent smell. Remember, so it's produce this kind of smell, a bit strong smell. Okay, not very nice smell actually, like a sulfur, stinky, and smoke smell. Okay, because of the production of decanal and nonanal. Okay. And then the second note should be more um, fresh smell. Okay, fresh smell. For example, like orange smell. Okay, the grassy after the rain. You know, that's kind of smell. Okay, that should have that smell for the perfumery industry. Okay, the one the second note should be that fresh smell. So Kesum also produce this fresh smell. Okay, so the compound that contribute to that is the dodecanol, carfilin, humulin, and tetradecanol. Okay. And then the last note should be the pleasant, very nice smell. Okay, and then it lasts longer, which is the third note. Okay, Kesum produce this third note as well. Okay, the fruity, the floral, pleasant, woody smell. Okay, is contributed by the dreaminol and bezafuran. Okay, so that's why make this company Firmenich is really interested to have the essential oil from Kesum because of this. They already have these 3 different notes for the perfumery industry. Okay, so something interesting, right? Because we never know that Kesum have this different kind of smell that we know is like pungent smell, very like not nice smell. Okay, but in Kesum we do have this smell, and we know this is the compound that contribute to the smell. For example, like dodecanol, we know that the smell of dodecanol grassy, pungent, orange peel, and soapy, like soap smell, very nice. And that's the carofilin also produce that kind of smell and so on. So from there we know that okay, this is the compound, the group of compound that contribute to the smell. Okay. So after we profile all these custom compound by using different technique, different metabolomics tools from GCMS, LCTOR, HPLC, and GCMS, we come out is about like more than 1,000 compounds. Okay, so by using the different tools. Okay, as I always said, metabolomics, you cannot do metabolic by using only single technology, only single technique. Okay, it's like a comprehensive technologies, comprehensive technique. You cannot say that, okay, we already have the TOR, we don't need other things. No, we need all the other system as well. If you said that we already have the GC, we don't need that. No, we need all the system in place for us to have the proper metabolomic study. Okay, so this is the potential compound. Okay, this is from the literature actually. These all compound that produce in kesum. Okay, so from there we know, okay, kesum produce the kind of anti-tumor, anti-inflammatory, anti-cancer, anti-malaria, and so on. Okay, that make us more excited, okay, to know more about the compound from kesum. Is it they have this kind of biological activity? Okay. Okay, from our previous work, okay, the one that I already profiled about more than thousands compounds, okay, there is a lot of unknown compound. Okay, there's a lot of unknown compound that need to be explored. Okay, so in this study, we this time we have two postdoc that time. Okay, the first one is Prof Sahidin is already professor at Indonesia. Okay, he's the first one that come up with the polygonomines A compound, the pure compound from kesum. We give name polygonomines A because it's a pure compound, a novel compound from kesum that's not been identified yet. Okay, so then from there we do the cytotoxicity study, antioxidant study, and anticholinesterase study. Okay, and then I have another postdoc. He just finished last 2 years. Okay, Dr. Andy Rifki, okay, from the ITB, Institute Technology Bandung. So he managed to come up with the derivative of polygonomines A, which is we name it polygonomines B, C, D, and E. Okay, so now we have 5 isolated compound from kesum. All these are pure compound and also new compound. Okay. Okay, our work has managed to publish in a scientific report for the polygonomines A work. Okay, we have test different biological activity for polygonomines A. So we know that, okay, polygonomines A have all these kind of properties. Okay, this is thank you to Puan Rafida for her excellent contribution to the works. Okay. So, and then this is the isolation of the compound. Is many steps of isolation of the compound. Okay, so this has been done by Prof. Saidin and also by Dr. Andy Rifki. Okay, so different fractionation and so on. Okay, it's not easy. It's not easy step. Okay, many steps. So we come up with that. Okay, so from there we have elucidate the structure. Okay, showing that our polygonomines A have a bit similar to venocoside A structure. The differences is only on the at the methyl peroxide and hydroxyl structure. That's only the difference. Okay. So and then we managed to patent this. Okay, our compound. Okay, is already filing patent that time. Okay, and then from this, we have come up with the anti-cancer properties for this compound. Okay, it shows that polygonomines A can be used to treat cancer such as leukemia, colorectal, and breast cancer with very remarkable activities. Okay. And then, as I mentioned, there's not much difference between this polygonomines A with the venocoside A. Okay, and then from the publication, from the literature, we found that there's only one publication in 1994 on the venocoside A and B, that they have tests on the protein kinase inhibitor. Okay, this actually polygonum Pennsylvanicum. Okay, it's not polygonum minus that one that we study. Okay, protein kinase inhibitor is to reduce the HIV activity. Okay, so we have identified the compound. Okay, and then we come up with the study on the molecular docking or the polygonomines A against the HIV, because from the information on the venocoside A showing that venocoside A have these properties of the anti-HIV. So we test first we test with the molecular docking. Okay, it shows that both of this compound, okay, can bind at the same pocket of the HIV protease. Okay, but by using the different orientation of the ligand bindings. Okay, so So it shows that, okay, polygonamins A also have these anti-HIV properties. Okay, so with my collaboration with the Chulalongkorn University, okay, we send our sample there, and then they test the anti-HIV by using their HIV protease enzyme. Okay, so it shows that ours also produce very good activity, okay, of the anti-HIV. Okay, so we know that, okay, polygonamins A also can have these anti-HIV properties. Okay, so then it's just like to conclude our finding on that, the study of the, because people know that kesum also have this smell, they have the anti something on the insect repellent. Okay, we come up with a simple experimental design, okay, to show that this kesum also produce this insect repellent properties. Okay, but in this study we use cockroach. Okay, we put our essential oil, okay, and then with a different dilution, okay, inside this aquarium, okay, proper aquarium, and then after 30 minutes, okay, the cockroach is run away from the petri dish that contain the essential oil. Okay, after an hour, or this cockroach already die. Okay, after a day, no more left. Okay. So we come out with the polynus TM oil. Okay, so we trademark this with the help of the PIK UKM to trademark this polynus oil, the essential oil of kesum. We call it polynus TM oil. Okay, so we already have the trademark. So if you want to use our polynus TM oil, polynus oil for your any type of your product formulation, you can contact me. Okay, so this is our, then we want some innovation that time. Okay, okay, this is our group, the Metabolomics clan at INBIOSIS. Okay, many with happy pace doing research on the Metabolomics. I think that's all from me. Thank you so much. Back to Dr. Murni.
Thank you very much, Dr. Sorry. All right. Thank you very much, Dr. Syarul Nataqain, for sharing us a very interesting topic. So, if there is a question from the audience, if you have, you kindly can type it on our chat box. Okay. So, Dr. Ain, for your polliness just now, is using essential oil, isn't it? Is it poisonous to other people, or how?
Essential oil, the one that you mentioned about the insect repellent, right?
Yes.
Okay. Because kesum produce the smell that, for example, the carophylline, okay, that we known as the insect repellent, okay. So that's why, since because kesum produce this smell, so it's good to use kesum for insect repellent. But for human, because we can eat kesum, right? So up to now, there's no any people that because eating kesum, but we are not suggesting you to eat the essential oil because it's very concentrated oil. Okay, very concentrated kesum oil. It's not meant for to be eat. Okay, but it can be used in other application. For example, insect repellent, fragrance industry, and so on.
Okay, thank you for that. I have a question from our fellow, Dr. Halida. May I know if you have done any biological study on different polygonal sample groups that show distinctive flavonoid biomarkers, Dr. Ain?
Okay. So actually, we did that study. Okay, there is a publication on that. Actually, okay, I'm not sharing here today, but there is a differences of the biological activities of different plant population. Okay, so that's why I can recall. Okay, for example, the one that produced in Highland. okay, the from the one that Fraser, Genting, and Cameron Highland, okay, they have more compounds, okay, toward the flavonoid, okay. The one that produced in the Uluyam and Genting, they have more compound towards the terpenoid group, okay. So these two group have a different properties, okay, because you know flavonoid is what for different properties, and then terpenoid also for the different properties. So it's distinguished between this population actually.
Okay, since you mentioned about different places just now, I have a question regarding the research design. You mentioned about using chamber, right? You take from 3 different places, Uluyam, Lenggeng, and as well as Genting Highland. And later on, you use chamber. What is the use of the chamber?
Okay, so we grow all these plants because we took the plant from the population, right? From the environment, okay? Okay, because the plants grow in the environment, in their own places. Okay, we took that plant, okay, and then we grow it in our growth chamber. So we can control that environmental factor that will affect the plant metabolite production. So we give the same temperature, the same humidity, everything the same, so the plant been treated as the same, so you can, that's why from the compound that they produce, that we know is not being affected by the environment.
Why it is significant to grow in one particular place? Why don't we just take from 3 different places and directly do experiment after that?
Okay, so because this study actually you want to know the temperature, okay, we play around with the temperature, okay, but if you take it from the environment, we don't know that the exact temperature, right? Because like sometimes if you take it during the rainy season, you will get a different temperature, okay, and then you take it during the hot season, we get a different temperature. So actually you cannot control what is the temperature, what is the actually the exact temperature, but while when putting in the growth chamber, so we know that, okay, the plant has been grown at this 20 to 24 degree and so on.
Okay, okay. Okay. Okay, we have answered the question from Dr. Khalida just now. Okay, there is one question from research fellow Dr. Nisha Govinda. The new compound isolated from Kesom were docked against HIV molecular targets. Okay, doctor, could you elaborate the opportunities of this compound as antiviral target? Any similar in silico study conducted against SARS-CoV-2 targets?
Okay, we never yet doing any study on that kind of virus. Actually, I'm excited, interesting if I know that if this pure compound also can have a target on that SARS virus or maybe COVID virus or something like that. But we never done any molecular docking on that yet. So maybe if Dr. Nisha, if you want to do that, I'm more than happy to give you the information.
Yeah, maybe Dr. Nisha can collaborate with Dr. Ain regarding the effect against COVID virus. All right. Yes, I have another question. Has metabolic approach being applied by industry in Malaysia, especially in agriculture?
Okay. As far as I know, because I have a collaborator with the industry, right? So, most of them, they are not doing the metabolomics study in their place, okay? Because if you want to do the metabolomics study, you need to have the system, right? So then they don't have the system in place, okay? But they send their sample to us. So we did the study on the metabolomics part at our place, and then we discuss on that. We give the result and so on. So from that study, we come up with the interesting biomarker for the plant disease and so on for the agriculture. So we have that kind of study as well, but with the collaboration. Okay.
So industry must collaborate with university to do this
Yes.
kind of approach.
Yes.
All right. There is a question. Yes. Rizwan Zakaria said he is looking forward to understand how OMIC can be applied and translated into industry. Maybe you can comment on that, Dr. Ain.
Okay. So as we are focusing on the metabolomics, right? Okay. So from the metabolomic, you can discover a biomarker, right? Okay. From the biomarker, there's a more information that we discover from this biomarker that can be applied to industry. Okay. So, for example, like the one that I mentioned about the disease study. So we know the biomarker that can make the fish survive, and also at the same time, increase the growth performance of the fish. Okay, the study is now conducted by my student. So we know that okay, this is can be applied to industry by maybe using it as a feeding formulation. Okay, maybe you can use it as a later on maybe as a vaccine or something like that. I'm not sure, but there is more opportunity to explore. For example, in a plant, in a agriculture study, so we know that okay, different temperature have a different effect of this plant. So and then we know okay, this is a biomarker that contribute to. Okay, then why don't you use this biomarker to be applied in the industry? So my study also involved on the Alzheimer study. So we working on the Alzheimer now. So we have found that compound, okay, the proncusum that contribute to the Alzheimer activities, okay, that can reduce the effect of this. I'm not saying it's toward the Alzheimer, but it's toward the cognitive. Okay, so we have something that can be explored with the industry as well. Yeah.
So you were saying just now, I am interested with the Alzheimer disease. So is there like a good advice for old people to eat more kesum?
Yes, you can eat kesum, but not eat too much. Everything is not good if you eat too much, right? So it's just
Yes.
very, I mean, the reasonable amount should be okay. Okay. Yes. Yeah. Because but the thing is that kesum have the very high oxalic acid. Okay, oxalic acid is not good for your kidney. Okay, so that one you need to consider on that. That's why if you eat kesum, you need to cook it, boil it, not eating raw
Ah,
what you call it, like a kerabu, something like
like
that.
ulam, something like
Like
that, right?
ulam, yeah. So better you cook it so the oxalic acid will gone. Okay.
I see. There is a question from research fellow Dr. Imelda. How do you maintain the unique flavonoid character if taken from the highland, for example, if have intention to mass produce in the plot?
Okay. Okay, that's why we need to know the temperature. We need to control on the temperature, but it's not that practical actually if you want to grow it in the plot, right? Because it's too many how to control the temperature because we don't have the glass house and so on. We just grow in the plot. But what I can say that is that we know okay this type of compound okay produced in this population. Okay, for example, it's produced in the Cameron Highland population. They produce a lot. Then we can bring this plant, the Cameron Highland plant, back to our plot, and then we explore on that. Okay, so there will have, of course, and we need to anticipate that the activity, I mean, the intensity of the compound will be reduced or maybe gone or maybe increased. We never know. That's why we need to study on that. That we need to explore more on that before we can grow the plant in the plot. So, okay, from our previous experience, uh, we use the plant from the ulu yam, okay, and then we grow it at INBIOSIS, and then it shows that the plant produce all the compound that we want. So it should be okay.
Okay, thank you for the answer. Is there any more question from the chat box? All right, I think no question. Okay, if there is no any other question, I think we have come to the end of the webinar. So, oh yeah, we have another question from Prof. Ibrahim. Challenges on accurate determination of metabolite using metabolomic approach, Dr. Ain.
Soalan otai ni.
Yes.
Okay. Okay. So there's a difficulty, okay, the challenge of that metabolomics, okay, to have the accurate identification of the compound, right? So yeah, in order for us to do that, okay, the best way that we need to have the MS, okay, not the only MS, but it's MS. So we can confirm, not really confirm, but we can have that. We can what you call it? We can remove the not true compound Okay, so we can have the proper identification of that compound. Okay, another thing is that by using authentic standard, but the problem with the authentic standard is not available in the market. Not all available in the market, right? Especially from the secondary metabolites. Okay, so another technique that we can consider is on the NMR technique. Okay, but back to the question. Okay, how much information that we can gather from this by using the NMR, because NMR also is another technique with the different structure elucidation and so on. But like the isolated compound is much more easier if you are using the NMR. Metabolomics for NMR also established nowadays. But yeah, it's back to how sure we are that the idea of the compound is correct. But until now, that's a challenge, Prof. Okay, maybe in the future we can ask the company if they can explore more how to get the identity of compound, the true identity of the compound. Yeah, but as for now, that's a challenge. So if you want to use the authentic standard, there's not many authentic standard available in the market. It's just based on the spectrum from the MSMS. That's what we can do up to now. Okay.
All right.
I hope that answer Prof question.
Okay. Thank you very much, Nataqain, for the answers. Okay, I think we have come to the end of the webinar. I would like to thank Associate Professor Dr. Syarul Nataqain for the very interesting research topic. And I'm sure this topic has opened up our view and give an idea on how you want to bring up your research output to the next level in metabolomic approach. Okay, today's webinar had recorded around